Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speakers in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion and I'll ask our speakers your questions. Your questions in the Q&A window will only be visible to myself and our speakers. So thank you for joining us today. It's now my pleasure to introduce our speakers, Andres Castillo and Shania Zhang, PhD, both portfolio managers from Sartorius.
Thank you, Alex, for the kind introduction. My name is Andres Castillo, and I'm super excited to be able to present today with my colleague, Shania. But before we get into some of these screening applications, I wanted to go over a few of the overarching challenges. So here we have listed the time to market and the cost of goods sold. And so we have a few key drivers here that make these such a challenge within the space. And so the first is that there are so many complex interactions within viral production. To assess them in such a short time is really difficult. So we need to find solutions in order to enable that. The secondly is that it's a relatively new space with limited personnel and established know-how. And so being able to collect that information as quickly as possible and leverage that is really essential. And finally, all the tedious revalidation, optimization while scaling. I think that it's imperative that we be able to leverage systems that make it easier and more robust in the initial collection in order to facilitate that scaling ultimately into production. So how do we begin to tackle some of these challenges? Well, at Sartorius, we've taken our wide portfolio of products and really created an ensemble that's application focused so that we can start to address some of these needs within the viral therapeutic space. So we begin by acquiring the information, right? We need a robust platform that can be able to handle all the culture conditions that you need to test, as well as the process parameters within it, in order to optimize and be able to generate a robust amount of data. Secondly, you need the tools to be able to analyze all that data and create meaningful insights in a way that's not only shareable and communicatable, but also allows you to progress further into the optimization process. And finally, it's the application, right? So this creates a basis of understanding, and so that you can then apply it to future projects and accelerate those timelines, ultimately reduce the cost as well. So what's in the screening ensemble? Well, the screening ensemble for viral-based therapeutics was really designed to integrate into your upstream workflow. And so you can use the screening ensemble to screen process conditions, identify CPPs and CQAs, and set the design space. So the end result being that you can achieve a master and a working cell bank for use in further bioprocessing. So let's jump into a little bit more detail. After choosing a stable expression or a transient approach, media can be screened for cell cultivation, optimal parameters. It can also be found for plasmid transfection or viral infection. Product characterization, cell quality attributes can also be gathered using the Sartorius bioanalysis set of instruments. And then all of that connects into the analytics system where further data analysis and insight generation can be done, and then future design spaces can be set. So using our design of experiments and multivariate data analytics products, you really have a powerful in-house data analytics suite that the whole lab can use. And so what we start out with is the base, right? And so in order to enable a high throughput multi-parallel experimentation, we have the Amber 15, which can run up to 24 to 48 experiments at the same time. And in order to do media screening, for example, you need high quality media reagents. And at Sartorius, we have the Excel portfolio of media that allows you to find the right fit of media for your process. In order to assess the quality of the process and to identify some of those critical quality attributes, we have a whole suite of bioanalytical software that instruments that we'll get into, such as the IQ, the virus counter, the InQSite, and the PathFix HPLC. And finally, how do we connect all this information? Well, we have MADE and SIMCA. So we have MADE to do the design of experiments, set the design space, and it intuitively guides you through being able to assess multiple parameters at the same time. And then SIMCA, which allows you to do MVDA, multivariate data analysis, and really connect all that bioanalytical information into your process as well. The ultimate goal, obviously, is to get those master and working cell banks. And so we have the fillet system and the testing services at the end to be able to generate those cell banks and make sure they're of high quality. So as I mentioned before, the Sartorius Screening Ensemble is really designed to fit into your existing workflow. So what do I mean by that? Well, let me just walk you through an example. So if you wanted to screen multiple media to find the optimum, well, we have a full selection of media and reagents. And in the same time, you can test those other culture conditions. And we'll get into some of that data in a little bit. All of that is guided by this MADE software and enabled by the multi-parallel bioreactor, the Amber 15. When we want to connect some of that bioanalytical equipment I mentioned earlier, we can use the SIMCA multivariate data analysis software to connect them. And so if you are running, for example, high throughput flow cytometry on the IQ3, you'll be able to connect that data and generate those insights. And finally, obviously, the ultimate goal within this process is to achieve those working in master cell banks. And so we have the fillet system and our testing services that really enable this. And so now I've gone over the overarching view of the screening ensemble, and I'm really happy to pass it over to my colleague Shania, who's going to go through some of this data.
Thank you, Andres, for providing a nice introduction to the Sartorius Screening Ensemble for viral-based therapeutics. Now, let's dive into some in-house data as well as industry case studies generated using Sartorius Screening Ensemble. As Andres introduced, your journey begins with vector design and the selection of media, reagents, and cell lines. Many hex cell lines produce varying levels of AB serotypes. For example, in Figure 8, two different hex cell lines were tested for different AB serotype production using a 2 plasmid AB system through PI max with a 1:4 ratio. The AB titers were measured by qPCR. As you can see, hex cell line number 2 consistently produced more AB titers for all serotypes than hex cell line number 1. Even with the same hex cell line number 2, the viral titer varied among different serotypes. Understanding such complexity of viral vector production, after testing 5 hex cell lines, 4 transfection reagents, 6 AB vector serotypes, and against 3 reference media, Excel, now part of Sartorius, has developed a portfolio consisting of 4 high-performing, chemically defined cGMP-grade hex cell line culture media and 1 feed for suspension culture to accommodate any viral processes. As shown in Figure B, Excel's HEC-VIP-NX medium constantly produce a significantly higher titer of AB2 than a commercially available reference medium in all 5 different hex cell lines. From these data, we can see that it's important to screen for multiple cell lines and multiple culture medium. We encourage you to try the kit from your own lab to select your best cell line and best media. Another important thing is to screen for the cell culture parameters for suspension culture, which is more suitable for future scale-up than adherent culture. To screen for suspension culture conditions, 3 stirring speeds, 3 pH, and 3 dissolved oxygen conditions were tested in parallel using Amber 15, the automated high-throughput microbioreactor system, which serves as scaled-down model, and the design of experiments approach with integrated MADE software. As shown in Figure A, 400 RPM, pH 7.3, and 30% DO yielded a viable cell density of more than 3 million cells per mil. pH and stirring speed were identified to be parameters that have a significant impact on cell growth. To further facilitate the interpretation, the data was also visualized in a contour plot. The predicted probability of failure for viable cell count, viability, and generation time were plotted based on different pH and stirring speeds. It's shown in percentage, with green being the lower probability of failure and red being the higher probability of failure. Since DO was not found to be a significant parameter, 50% DO was chosen as a set point. From the contour plot, we can see the optimal set point is with stirring speed at 400 RPM or less and pH being approximately 7.22. Of course, viral vector production is a more complex process, and we can continuously utilize EMBER-15 and MODE to further screen for more process parameters. As shown in this case study generated by the University of Nantes, they used EMBER-15 and MODE to screen for multiple process parameters such as cell line, median, transfection reagent, total plasmid amount, and complex time. The intuitive MODE software can automatically rank the process parameters to show which parameters are critical to the AAV titer. The linear regression coefficients from the initial screening design shown in Figure A were used to assess the importance of different factors. The bigger the bar away from the center zero line, the more impactful the factor upon the targeted response, in this case, viral vector titer. Also, positive coefficients increase the titer, while negative ones decrease the titer. As a result, they were able to select the factors having the bigger impact on the titer increase, and then narrow down the scope to further optimize the AAV production. The titer regression coefficient obtained from the optimizations design shown in Figure B are more accurate compared to the model from the initial screening design and support both linear and nonlinear terms. As a result, cell line, media, and transfection reagent with total plasmid amount were identified as critical process parameters, CPPs. These case studies were used for AAV production. But what about lentiviral production? On a similar note, our industry partner Oxgene, now part of WuXi AppTec, used the same approach to screen and optimize their lentiviral vector production. As you can see from Figure A, instead of using the intuitive screening and optimization approach, where all the variables are tested one after another, they utilized EMBER-15, leveraging automation and high throughput, and a design of experiment DOE approach with our integrated MODE software for screening, optimizing, and validating multiple process parameters simultaneously. Only 2 DOE runs were needed for the optimization of 9 parameters, with more than a 50% reduction in the total process development time. As a result, in Figure B, you can see the first round of DOE facilitated a 10-fold increase in infectious titer compared to the initial process. Those data informed additional improvements to cell density and media components made in separate experiments, shown as intermediate conditions. Finally, optimizing the process parameters during the second round of DOE delivered infection titers up to 34-fold greater than the initial conditions. With the high cost of transient transfection associated with the GMP-grade plasmid DNA, people are also investigating stable producer cell lines, which don't require transient transfection. Here's another case study from GSK in 2020, where they used the EMBER-15 system for rapid lentiviral vector producer cell line generation. And in order to rank the 12 cryobank clones resulted from a cell line development campaign, lentiviral vectors were produced from 12 clones in duplicate in the 15 ml single-use EMBER-15 vessels. And then the unconcentrated physical titer was assessed by ddPCR, and the functional titer was assessed for P24 by ELISA. The functional titer of the highest expressing clones, clone number 3, was more than 1.5 times 10 to the 7 transducing unit per mil. EMBER-15 facilitated screening conditions for 12 clones to obtain a stable producer cell line in parallel, with the benefits of reduced production costs since the experiment was run in a 15 ml scale. Furthermore, the screening studies on EMBER-15 enabled initial runs on 50-liter scale in single-use stir tank bioreactors, suggesting this process was transferable from 15 ml to 50 liter. I've gone over a broad selection of applications that the screening ensemble can support in viral vector production. Now I will hand it over to my colleague, Andres, for some final thoughts.
Thank you so much, Shania, for going through all the applications in the data. We can really see that the Sartorius Screening Ensemble is a really powerful system to first, as I mentioned before, acquire a robust set of data. So we've been able to survey more in less time with this multi-parametric screening. Then we were able to analyze it through unique tools and insightful graphics using our software suite, Made and Simca. And finally, we were able to then build upon that knowledge. And as we've seen in the last example, really move towards scaling. And there we have a whole process knowledge ensemble that can really guide that. And so I just want to thank you so much for joining us today, and we'd be happy to answer any questions.
Okay, great. As a reminder to our audience, you can go ahead and type in your questions for our speaker now. So our first question is, what features do you look for in media for optimal HEK cell growth and viral vector production?
Yeah, I can take that one. That's a great question. So first off, chemically defined, xeno-free, and suitable for suspension culture, and that really ensures reproducibility. And next, you want to take a look at does the media fit your process? So is it scalable? Does it come in flexible formats? And is it fit for fed batch or perfusion? But importantly, you really need to screen the media for different stereotypes and cell lines. So for example, Sartorius, the XL media portfolio consists of 4 high-performing HEK media designed to fit within the process. And we would encourage you to try them out so that they fit the transfection reagents or the cell line or any viral vector process.
And why is it important to use an orthogonal analytical approach?
I can take this one. In January 2020, the FDA published several regulatory guidelines for cell and gene therapy. And in case of viral vector analytics, the changes were made for identity, strength, potency, purity, safety, and stability. So these analytical methods require validation before they're acceptable for the use in the clinical manufacture. So their usage in research and process development is not only important to improve the manufacturing process, but also to validate that these methods can satisfy the regulatory demands. So the key to this will be the usage of parallel orthogonal methods with appropriate controls to ensure the integrity of the data. So it will help assess the viral production, and also help develop the best process. Another important thing is that their speed, accuracy, and robustness is important so that we don't need a lot of sample processing. And at Sartorius, we offer several high-throughput automated analytical tools. For example, IQ for transfection efficiency, infectious titer, InQSight for live imaging to assess the infectious titer, virus counter for lentiviral particle quantification, and also PASSIX HPLC for lentivirus packaging, and Octet for AAV quantification.
Okay, so it looks like we're running short on time. If we don't get to your questions, then we'll be passing them along to Andres and Shania, who can follow up with you directly. So please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is, how would you select a good scale-down model?
Yeah, I can jump in here. So obviously, a scale-down model is really just a tool for cost-efficient development and characterization of the processes. What you really need is the space to test all the parameters and the inputs that you want to test. You need the consistency as well, so maybe relying on automation within it, and then you also need the ability to measure the parameters in the process that you're developing as well. And so whatever scaled down model you choose would hopefully mimic what's at the manufacturing scale, but just for cost reasons at a small, obviously takedown scale. So for example, we have the Amber 15, which is a high throughput microbioreactor, which can do 24 to 48 parallel experiments at the same time. And so really leveraging that consistency and then building that through geometric consistency throughout really helps you build that identify those critical process parameters and critical quality attributes.
Great. Well, thank you, Andres and Shania, and thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.